Hello everyone, my name is Anthony Côrtes, and today I'm going to present to you guys the paper I select for the pesticide resistant class, which is gene amplification confers glyphosate resistance in Amaranthus palmeri. So the adoption of transgenic crops had been rapid since the technology introduction, and especially after the introduction of the glyphosate crops, the glyphosate resistant crops. Glyphosate was widely used in agriculture. That's happened because the glyphosate was good enough to control every weed in the field, and the glyphosate was exclusively used on multiple applications each season in a monoculture system. But because of that, without a mode of action rotation, without the use of other herbicides, there was an increase in the selection pressure for glyphosate. So as we can see here in this graph from Weed Science Society, we can see that after the introduction of glyphosate resistant crops, there was a huge increase of glyphosate resistant weeds around the world, and Amaranthus palmaris was one of them. So if you think about this weed, since 1997, azole was the main tool for Amaranthus palmaris control, and back in 2009, there was already um, estimate of 250,000 hectares of Amaranthus palmaris resistance, so which is a lot. So when we think about resistance and we think about this weed in specific, so Amaranthus palmaris is problematic because it's already a very aggressive weed, if I can say that, because it has a very fast growing. It's an annual broadleaf weed. So this plant can grow for around 2 or 3 inches per day, and commonly reach like 7 to 8 feet. And as you can see, also this plant has a high water use efficiency and a large quantities of seeds, around 250,000 per plant. So this weed, Amaranthus palmaris, is competing to resource with the crops that we have. So if you look to that, we can have yield lose around 91% in corn and 79% in soybeans. And which make this weed worse is that a lot of populations have developed resistance to multi-class of herbicides with different mode of actions, and one of them is the glyphosate. So it's important to us to understand this paper and to figure out what's going on. It's also important to understand the APSPS mode of action. So when glyphosate is applied, it blocks the enzyme APSPS, which catalyzes the bind of the shikimate 3-phosphate with the with the pyruvate, and these inhibit the production of the next compound, which is the enol-pyruvyl shikimate 3-phosphate, and this leads to a high accumulation of shikimate in the plant, and also interferes in the formation of aromatic amino acids like phenylalanine, tyrosine, and tryptophan that are very important to to protein synthesis and also to the secondary metabolics. So it's a problem. And when you look through and you also need to understand how a herbicide works inside the plant. So first of all, when you spray a herbicide, there is a penetration of the molecule inside the plant, and then translocation to the target protein. and then the accumulation at the target protein, and the herbicide molecule will bind to the target and then interrupt the metabolic and physiological process that will lead cell and plant death. So plants develop resistance, like you can have reduced penetration of this herbicide. You also can have altered translocations, like this herbicide, this molecule can go to the vacuole, or we can have the alteration in the gene of this protein change the characteristic of the protein, so the molecule can bind anymore to the target side. So it's a kind of resistance, target side resistance. Or we can have a target overproduction, so we have like overproduction of the target side, so the dose that would kill the plant is not working anymore. So it's important to understand that, to understand the next step of this paper. So because of that, the study objective was to use was to use genetic and molecular analysis of APS-PS genes and protein from glyphosate resistant and susceptible Amaranthus palmeri population, and demonstrate that amplification of the APS-PS gene is the glyphosate resistance mechanism. So, as a plant material and genetic population, seeds from resistant palmeri were collected from a field site in Malcolm County, Georgia, and azo seeds of susceptible palmeris were collected from University of Georgia Pond Farm Research Station. So, all the seeds were germinated and transplanted into large pots of growth in a greenhouse, and the resistant phenotype of which plant was confirmed using an in vivo liver disc assay. And also the R mods were placed next to the S females to create a F1 generation. And after that, this F1 was sprayed with a low rate of glyphosate to select for heterozygous resistance per gene. So one resistant F1 male and one resistance F1 female to generate the F2 generation. They were hand pollinated, and also the same happens to open pollinate to generate open pollinate F2 by placing different R female and male half siblings from the F1 generation. So the first step of this paper was actually to test the APS cDNA sequence, and for that the EPS-PS sequence from R. tuberculatus was obtained and used to design the primers for palmeri. And the primer sets were used to amplify the fragments region of the APS resistance and susceptible gene. And after that, there was a PCR reaction with 5 minutes at 94 Celsius, followed by 30 cycles of 1 minute, and at 94 Celsius, and 1 minute at 58, and 1 minute at 72, with a final extension of 10 minutes at 72 Celsius. So 7 R individuals and 2 S individuals were taken and used to make the APS F1 and APS L1 PCR product. So the PCR product after that were ligated into plasmids, and the plasmids were transformed in Escherichia coli cells. And after that, the plasmids were isolated for SANG sequencing with the M13 forward and M13 reverse primers. So after that, with the sequence, the sequence were alignment with the other plants APS-PS sequence gene to compare this. And here we can see that the Amaranthus palmeri resistance and then the Amaranthus palmeri susceptible, there was no difference between the alignment of the sequence. So because of that, you can see that the you can see that the case here of this project is not about target site mutation, which makes space to think about the amplification of this gene. So we can move along here. So for the next essay, effect of the glyphosate on APS, PS, cDNA, and shikimate levels were tested. So 5 plants each of R and S were sampled for 4 millimeter leaf disc for in vivo measurement of shikimate assay and RNA restriction before the application of the glyphosate and 8 hours after the treatment. So for plants R and S which were treated with glyphosate and water as control. So 8 hours after treatment, plants sampled again for 1 leaf disc shikimate and 1 for RNA extraction to test the APS-PS cDNA expression level. So the APS-PS cDNA was measured relative to ALS using quantitative real-time PCR. So here we can see that this table, and with a look up for the shikimate concentration inside the plants, we can see that the expression of EPSPS, we can see that the expression of EPSPS cDNA in glyphosate resistant and susceptible palmeri is not affected by the glyphosate treatment. So if you look here, comparing to the RLS gene, we can see that after the application there was no change, but even though we can see that the resistant Amaranthus palmaris has like 35 more copies than if you compare with the susceptible. And here in the shikimate, looking to the concentration of the shikimate, we can also see that after that the glyphosate was sprayed, the concentration of the shikimate increased in the susceptible plant, but not in the resistant plant, which means that actually improve the resistance about those plants, because as you saw some slides before, when we have the inhibition of the APS-PS enzyme, there is a high concentration, an increased concentration of shikimate inside the plant. So moving along, to test the APS gene copy number, 54 plants of each plant of the F2 generation and 12 plants, each in R and S were grown to the 4-leaf stage, and 1 leaf of each plant was used for an in vivo leaf disc shikimate accumulation assay with glyphosate, varying into 0 until 250 micromoles. So a shikimate curve was used to see the accumulation above the background level, and then 1 leaf from each plant was sampled for genomic DNA extraction and 1 leaf for RNA extraction for genomic APS-PS copy number, and also APS-PS cDNA expression. So after that, a quantitative PCR was used to measure relative genomic copy numbers, comparing to RLS in R and S individuals. So with that, we can see here this graph when you have like APS relative genomic copy number, and we can see the difference between the susceptible, that the number of copies was about 1 and 1.3. So for the susceptible plants, we can see a high accumulation of shikimate shikimate, sorry. And for the plants that had more genomic copies numbers of EPS-PS, we can see that the concentration of the shikimate was low. Here we have like one plant with around 5 copy numbers, which is not so many, it's not so much comparing to the susceptible, but still, even with hold of 5 numbers more, there was a low accumulation of the shikimate inside this plant. So azo was tested to see if this trade was irritable inside the plant. So through all these populations, like the hand pollination and the open pollination population F2, were tested for this to see if this trait was segregating inside the plant. So for both F2 generations, like the hand pollination one generation, the F1 mild parent had like 18 relative EPS-PS copies, and the F1 female parent had like 39 relative EPS-PS copies. And here in the graphic A, we can see the F2 hand pollinated. So we can see that this trait is actually segregating, because if you look at the frequency here, we can see that we have plants with one gene cop, but azo with 2 and 14 until like more than 80 relative genomic copy numbers. So we can see that this trait is segregating for both F2 generations, as the same is happening here for the open pollinate plants. So we can see that is happening here too in the B graphic. So yeah, and moving along, as the APS transgenic correlates was tested to see with the APS-PS genomic copy number. So select individuals from the F2 and parental populations were measured for APS transcript accumulation using quantitative real-time PCR. So yeah, as you can see, the APS relative genomic copy number has a strong relationship with the cDNA expression level. So if you look here, the circles, the white circles, empty circles, they are the susceptible plants. We have a low genomic copy number with a low cDNA expression level. When you look around to the resistance, the black circles, we have more genomic copy numbers, and also more expression of cDNA, EPS-PS cDNA. And when you look to F2 populations, we can see that we have something in between, like the information, the trait is segregating, but it's also correlated with the number of copies of the PS gene with a higher expression of the cDNA levels. So going on, as it was tested, the PS quantity and activity correlate with the PS genomic copy of number. So for that, young expanded leaf tissues was sampled from select R and S and F2 plants for protein extraction and EPS-PS quantification. So continuous assay for inorganic phosphate release was conducted with phosphate detection kit for assay for EPS-PS activity, And the APS-PS protein quantity was measured with immunoblotting. The dose response analysis in R the dose response analysis was in R, used to calculate the IC50 to the glyphosate and see which dose was able to reduce the APS activity by 50%. So if we look to the first graph here, we can see that the quantity of APS protein is deeply related with the relative genomic copy of number. So the much more we have and the much copy we have more quantity of APS protein inside the plant. So we can see this here, like the resistant, the F2, and the susceptible here in the bottom of the graph. So we can see that. And if we move along to see the APS activity, we can look into this graphic here in the bottom. So this graph is more about like the circle, the black circle, it's about F2 populations. Actually, all these 4 plants here are F2 population plants with different number of copies. So the first one here, this black circle, has around 54 copy of the APS gene, followed by the empty circle, which is around 39 number of copies, and the triangle black one, it's around 8 copies, and the last one is about 1 copy. So as you can see, the more number of copies of the APS gene, the more APS activity we have in the plant. But there was no difference between the IC50, no statistical difference for the IC50 inside those plants, which means that doesn't matter the number of copies, all the APS inside these plants, doesn't matter if this plant has like 50 copies or 1 copy, all the APS are sensitive to the glyphosate herbicide. So yeah, and to finish this paper, ESO was verified the distribution of the amplified APS gene in the genome. So what was used for that was the fish method, that is like a fluorescence inside hybridization, actually that. And this technique, it's about a molecular biology technique that's used to localize the presence and or absence of a specific DNA sequence on chromosome. So in this technique, a labeled DNA probe is hybridized to the chromosome, and the probe location is visualized under fluorescence microscope. That's what happened. So for this research, for this technique and methodology, the probe was synthesized using the EPS-F1 and EPS-L1 primers from resistant plant cDNA, and this cDNA was cloned, sequenced, and then PCR amplified from the plasmid, and 1 kb fragment from the EPS-PS gene was used at this fish probe. So here in the A, we can see that in the figure, in the picture A, we can see a somatic metaphysi chromosome of the glyphosate-resistant plant, palmeri, and in the picture B, we can see fish signals from the APS-PS gene probe, and in the picture C, we can see the merged image from A and B dispersed signals. For D, we can see an interface nucleus of glyphosate-resistant palmeris, and for E, a signal from the EPS gene probe, and in F, a merging of them. So what does it mean with all these pictures, what's going on inside the chromosome? So with these results, we can really see that we can suggest that the amplified EPS-PS gene were randomly found into the genome, like they are there causally. So moving along, and to finish the interpretation of this article, for me, this paper demonstrates that glyphosate resistance in the present wheat population is due to the EPS-PS gene amplification. It was solid results, and the increase of EPS-PS gene copy number is correlated with the EPS-PS mRNA, increased EPS-PS protein activity, and glyphosate resistance inside palmeri plants. The effect of additional EPS-PS copies is additive. Like, this is kind of additional copies confer high level of resistance. So this way, more copies you have, more resistance this plant is actually. And this gene amplification is not due to genomic duplication, considering the last methodology that I was talking with you. So, but this paper lets a question, but how does this amplification occur and is maintained inside palmeri resistant plants? So moving along and looking to other papers, like the papers that were suggested by Todd, there is this paper, the most recent paper about this topic. So second to this research, we can see that what they tell us is that an increase in gene copy number is actually extra chromosome elements that usually contain replication extra chromosome circular DNAs. So amplified EPS-PS copies in glyphosate is represented in the form of extra chromosomes DNA. So this is happening inside the plant? Actually, these extra chromosome DNAs are transmitted during cell division in mitosis and meiosis to the soma and the germ cells and the next progeny, the daughter cells. So, but this process, it still unknown. So we don't know how this is happening and how this actual process is happening, but the authors of this other paper that I'm talking to you now, they suggest that the dcDNA, it's an element and component of McClintock's postulate innate system. It's the hypothesis behind it. So for the discussion points, I was thinking about some questions that we can discuss later. So I have some 6 questions and discussion points for you. Like, do you think this article presents solid results? Do you think the methods were adequate to the study objective? And also, how do you see the applicability of these results for pest resist management? Did you have difficulty to understand any point in this article? And what new questions come from this research? And why APS-PS transcript abundance was measured relative to RLS gene? And why was important to have 2 kinds of F2 generations, like the hand pollination and the open pollination in this research? So I would like to say thank you to you, and yeah, I meet you guys in the discussion board. Bye.